It is my pleasure to introduce Professor Ian Frazer today for our interview. Professor Frazer is currently the chair of the Translational Research Institute Founding Board here in Brisbane and was previously the founding CEO of TRI. We all know Professor Frazer for his work together with Professor Janju on the development of the HPV vaccine against cervical cancer. Although your achievements need no introduction, I would like to mention a few awards, such as the 2005 CSIRO Eureka Prize for Leadership in Science, Queenslander of the Year and Australian of the Year 2006, the 2008 Prime Minister's Prize for Science, a Fellow of the esteemed Royal Society of London, the Companion of the Order of Australia in the Queen's Birthday Honours in 2012. We are really delighted to have this opportunity, Ian, to get your advice on future leaders for science. So this interview series, we are speaking to some of Australia's top scientists to highlight the challenges and the needs for scientific data management under the banner of the ARC Centre for Information Resilience. So may I start, Ian, by asking you to tell us a little bit about the types of data that you have used in your work and some of the opportunities that have arisen from the use of that data.
Every part of research that we do generates data. Without data, there is no research. So that over the course of my career, I've worked through clinically focused research where we collect information about individual patients or groups of patients. I've worked with technology to look at the sorts of cells that go around in the blood or the sorts of cells that invade a cancer, and we characterize each of those cells and collect data about each of them. So that from when I started working, we have always been collecting data sets. What's changed is that the data sets are getting more complex. With the ability to look at the genetic information now that we have, we're interrogating individual cells to look at what the properties of that individual cell are, and that might include looking at the expression of every single gene that's being expressed in that cell, so that by the time you take the 20,000 genes in the human genome and then take a sample of 1 million cells, you have a huge amount of data to look at. And therefore, managing that data is critically important for getting sensible answers out. You've got to ask sensible questions of the data set in order to get good data out, but at least we recognize now that almost everything that we do is data driven, and therefore we have to be able to handle large data sets appropriately in order to get quality results.
Mm-hmm. Absolutely, no science without data. And, with the collection instruments, and we see it, of course, in biomedical science, we also see it in social science, that those instruments for data collection are pretty robust and they've been around for a while. But once that data is created, and especially the large-scale data, tell us a little bit about the challenges in getting the data ready for analytics or the curation processes of that data.
Well, obviously it's important to get quality data, and therefore quite a large part of what you spend your time doing as a scientist is checking that whatever you're doing is in fact generating quality data. One of the most important measures of that is reproducibility, of course. If you can do the same thing twice and get the same set of answers out of the system twice, you've probably got a quality data system. But it's also important to remember that there are a lot of things that you can't control for that will vary across the experiments that you do, and therefore you have to look out for the variables that are not in your control that will impact on your data set so that you get reliable data out. That's particularly important for clinical work where you really can't control all of the variables round about a particular problem. You might be very interested in how a drug impacts on the development of a disease, and you'll get a data set about how the disease is developing. But if you don't also account for the fact that there are other things outside of what you're doing that are going to impact on those data sets, even simple things like diet, the weather, how the person's feeling that day may impact very significantly on the data sets that you gather. So that then requires you to average over large numbers of individuals and average over time and space in order to ensure that the quality of the data is good in the sense that it's actually there to answer the question and not some spurious outside influence that's making your data less good.
Yes. Yes, indeed. I mean, there is a lot of data out there, and some of that data is created through the scientific processes, and indeed the generative process and the robustness in it is extremely important. But some data is simply out there, and you mentioned about lifestyle and weather and different. What has been your experience in trying to link different data sets together, and the challenges faced by the scientists in sort of repurposing different data?
Well, there's always a risk when the data is already there, because you don't have control. And as they say, garbage in, garbage out. So you have to make sure that the data set that you want to use is valid. And that validation process is really challenging, because if you didn't generate the data set, if it's already there, it's quite hard to go back and test to make sure that it was generated in a reasonable way. And it might have been generated for a completely different purpose from the one you want to use it for. Indeed, that's very commonly the case. You get a data set on a group of patients, and the data set were generated just in the routine care of that patient. But you actually want to ask more of those data sets than just what was necessary for routine care. And the error factor, if you like, in the assay of the data for routine care might be quite different from the error factor that you would be prepared to accept on your data set. You might be interested in blood pressure, and if the blood pressure is relatively normal, people will take it and say, okay, it's 120/80 or 130/90, but you might actually be really interested in 2 millimeter changes, not 10 millimeter changes, and therefore you don't know whether your data set's been accurately gathered in respect of blood pressure that way. And you have to therefore go back and find out. So actually querying the way the data was generated in the first place becomes as important as querying the data itself.
Absolutely. And in terms of data partnerships and data sharing, so we know that there is, both nationally and internationally, there is a lot of movement in allowing data to be shared across jurisdictions, certainly in the scientific community. What has been your experience in your area?
Well, we know that we have to share our data. Yes. I mean, when we put together a scientific paper, which relies on a large data set that we've gathered, let's say we were doing single cell sequencing, RNA sequencing in single cells, so we've got all this data about the amount of a particular messenger RNA in a given cell over a very large number of cells and over an experiment. We get a huge data set out. It is mandatory that when you publish it, you also put that data set somewhere where other people can get to it. It's equally mandatory, but not often enforced, that you've got to specify more about the conditions under which those data were gathered, because that might be what you did might be quite fine for what you're wanting to ask of the data set, but other people will access that data set not knowing exactly how the experiment was done. It's as simple a thing as saying it was done on this particular machine using these parameters to set the machine up. And if that is appropriate for another question to be answered, that's fine. But if it's not appropriate, then unless you convey that information that it might not be appropriate, people will use it for the wrong purpose. So there's always this process of, well, how did they get that data set? How did we develop the data set? And how much can we really share? How we have to share the data. I mean, and the means by which we've analysed it. But the actual way we generated it, we put a rough description in there about how it was generated, and we hope that that's enough for other people to make sure it's valid for their purpose.
Yes, and that metadata question, I think it's been around in our field for quite a while, but with open science, open access, open source as in code and open data, the question of metadata keeps coming back, and indeed at this time there is a lack of it. And there is a lack of standards around which those metadata are described and presented. So that's a very good, very important point.
Yeah, it's a challenge for us really, because I mean we use other people's data sets and we expect other people will use ours. if nothing else, to validate the conclusions that we've reached from the work that we've done. I mean, it's not valid until someone else has found the same thing, at least not in my opinion. One swallow doesn't make a summer when it comes to data collection. So that you really want people to be able to go out and check that it's the same results that they get as you get, and it might be as simple as the software that they use. I mean, all these software packages that we use change all the time, and they get improved, which is fine. They meet the purpose that you want to use them for better than what was there before, but they might not be relevant for somebody else. In fact, for them, it might actually make the data less comprehensible or less consistent, and so that it's not just the data, it's the way you use it that counts. Metadata is important because it tells you how the thing's been generated, but the actual software packages themselves should really be deposited, and then you've got to say which version it was and did you alter it. And by the time you put all that together, it becomes extremely complex, and conveying that to somebody else is actually the big challenge in all of this.
Yes, I see that point. Very good. To wrap it up, Ian, I would really love to hear your thoughts on what are the needs and skills of future leaders. So we know that scientists, from all areas, in fact, even business, are working in data-intensive environments. So for our next generation of students, our future leaders, what sort of skills would you like to see?
Well, look, people should stick to their own area of expertise at one level, so that if you're really good at programming, then you should be really good at programming. But if you're going to use that programming to help a biologist understand a data set, you also have to understand enough about the biology that you can make sure the programming will do the job. And conversely, if you're the biologist and you're generating the data, you have to be aware of the limitations that the person who's writing the code to process your data will encounter in doing so. So it doesn't mean that every biologist has to be able to write program. I personally think that's a bad idea by and large. But I think that you have to understand the limitations that are imposed on the processing of the data by the fact that you're dealing with very large data sets now, and you may take shortcuts, if you like, and random sample into a data set rather than take the whole data set, simply because the computational power needed to control the whole data set is much more than you can get your hands on. So then you do random sampling, and therefore the programmer both have to understand what the limitation of that will be from interpreting the biology and from getting accurate answers out from the data set.
We totally agree with this collaborative approach. I think it's a team sport. I often talk to my data science students and we say it's a team sport. Be prepared to understand the domain in which you are working, be prepared to work with programmers and software engineers and with statisticians, and that's how we solve these big problems.
Yes, it's a collaboration, and it will in the future increasingly be a collaboration, and increasingly we'll have to have a bit of the skills that the other people around the collaborative table have.
Yes. Thank you so much. That was really helpful and very valuable. Thanks for your time, Ian.
Well, thank you for asking, because I think it's such an important question. As I say, when I started in science, we didn't have big data sets. And we didn't have to worry about that too much. N of 1 was fine, thank you very much. But now it doesn't work that way anymore. And the young people coming up through the system have got to understand that manipulation of data is going to be a key part of whatever they do.